Good morning, everyone, Francis, and everyone in attendance. It's really a great pleasure to be here this morning, and especially to be asked to kick off this conference, although like most people, I would have preferred to have been in Montreal in person. So what the topic is for this morning's lecture is the use of computational analysis in a setting of pediatric cancer precision medicine that we've had going on over the last 4 years here at our hospital in Columbus, Ohio. So my slides have decided to take off on their own. Sorry, so this is just my disclosures prior to the talk, most of which are not relevant to the subject presenting today. I want to start out the discussion today by just talking about the reality of adult cancers, where genomics is increasingly providing a treatment playbook. And what you can see here on the left-hand side of the slide is really how genomics has enhanced our understanding of non-small cell lung cancer and the different genetic drivers of this disease over time and over large-scale cancer discovery. And coincident with this increase in knowledge about drivers has been an incredible increase in the number of corresponding targeted therapies that can address many of these drivers successfully. And this has resulted in this diagnostic paradigm that currently plays out in much of adult cancer medicine today, where a diagnosis is performed on a small tumor piece from a biopsy needle or from a small resection. Genomic analysis takes place. And in the setting of lung cancer, for example, the presence of an EGFR mutation would invoke the use of a receptor tyrosine kinase inhibitor, a small molecule therapy. If EGFR and KRAS mutations are found together, this is typically now going to immune therapy with or without associated chemotherapy treatment. And in the case of these fusion drivers, such as ALK and ROS fusions, there are numerous inhibitors, again, of these particular drivers. And so I want to use this to set the contrast, if you will, between adult cancer medicine and where we're at in pediatrics nowadays in terms of clinical genomics. Mike Berger and I wrote this review a couple of years ago for Nature Reviews Cancer Clinical Oncology, rather. Again, just sort of setting the stage for how integral genomics is in a setting of adult cancer medicine, with now information clinically coming from somatic variants as well as germline variants, including new aspects of immunotherapy. But the reality in the pediatric setting is shown here. Unlike adult cancers, pediatric cancers typically have a low mutational burden, and they yield few, if any, druggable targets from just DNA profile alone. Clinical trial setting, or even with FDA-approved drugs, it's very difficult to dose pediatric patients properly, and there are many unknown adverse event profiles. As we're fond of saying in pediatrics, kids are not just small adults. Clinical trial enrollment often does exclude pediatric patients. This is changing a little bit in the United States with new legislation that's kicked in this year in 2020 that incentivizes pharma companies to open pediatric arms on many clinical trials where the targets are also present in the pediatric patient setting. And then cancer immunology, which I'll talk a little bit about today in a pediatric setting, is really not well understood. And in fact, we don't typically think about the use of immunotherapies like checkpoint blockades in the pediatric cancer treatment, although there are CAR T therapies that are now approved, which is one other type of immunotherapeutic. So the challenge in pediatric cancer, of course, is that it is, although a rare disease writ large, still the leading cause of death by disease in the developed world in children over 1 year of age. So it remains a significant health problem. And I would argue, as I often do, that even in the challenge of targeted therapies I'll talk about today, really what we want to do is get children more to these targeted therapy intermediates and immunotherapy intermediates in terms of treatment, as opposed to aggressive chemotherapy and radiation therapy, which, if you're lucky enough to survive pediatric cancer, predicts a lifelong high level of health-related problems because of these therapeutics. So we've had a large role in pediatric cancer discovery in a 5-year project that was done in collaboration with St. Jude, sequencing over 800 tumor normal genomes using next generation sequencing. And this was sort of a summary paper in Nature Genetics in 2012 that really brings home the points that I've somewhat already made. First of all, pediatric cancers are typically harboring very few point mutations. Conversely, we find that they have many common fusion gene drivers, and I'll talk about that more today. Indeed, it is the case that in most pediatric cancers, there are alterations at the epigenetic level, either to histone proteins that effectively change chromatin packaging, or other epigenetic drivers such as altered methylation. And the downstream result of these epigenetic drivers is that there's a modified transcriptional program that drives cancer onset and progression. So I'll talk a lot today about these challenges in the epigenetic setting and how really we're studying RNA to largely understand what they impart. And then lastly, the other finding from this large-scale study is that the pediatric cancer predisposition, namely germline susceptibility, has a much higher component in pediatric cancer than previously thought. So I'll talk about all of these aspects as we go along. This is just another paper from Nature a couple of years ago, not our work, but really reinforcing on the left-hand side of this slide the grave differences in mutation prevalence in pediatric cancers compared to adult cancers. The pediatric cancers are on the far left, and this really focused on significantly mutated genes. Again, in the comparison between the two, you see the Venn diagram on the right showing that there's very little overlap in significantly mutated genes. And in the gray area, many of the significantly mutated genes that are shared are actually tumor suppressor genes. And so these are very difficult, if not impossible, to drug. So in the setting on our new Institute for Genomic Medicine, one of the first studies that we opened was this cancer protocol, currently an IRB approved protocol using tumor normal exome RNA sequencing and methylation profiling, as I'll talk about today, across patients with rare and refractory relapsed cancers. This is the workflow overall, starting in the upper left-hand part of the slide, where we've really made it quite easy for the providers to nominate patients onto the protocol. This is all in the setting of REDCap, as you'll see in some of the bullet points at the bottom of the slide. And once we have a completed nomination form, our study team reviews that and either votes to accept or decline the patient's enrollment onto the trial. And this sets in motion a number of events that are played out throughout the rest of the slide, including consent, obtaining samples, and then analyzing the data, all of which I'll talk about today. I want to point out that our REDCap database records all the steps in the process. We do have de-identified information and analysis results also available in a Confluence site. And the REDCap connects the EPIC electronic medical record, or into the EPIC electronic medical record, I should say, and into the radiology imaging database so that we can see information about the patients in the REDCap environment that we don't necessarily store in REDCap. So this now just is showing an overview. I've already talked about some of these aspects from specimen intake all the way to interpretation and reporting. The eligibility, again, is around patients with rare relapsed or treatment refractory cancers, and some rare hematologic diseases have also been studied. We're doing what I call enhanced tumor normal exome, where in the pediatric setting, we get a lot of information from chromosome gains and losses, either focal or arm level. So we really have in the mix here, not only the ability to capture all genes, but also the ability to pull down sites along each chromosome at about 30 kb resolution. This gives us reasonably high level copy number information. We're typically aiming at about 250-fold coverage depth on tumor and normal DNA. And as I'll spend a lot of time talking about today, RNA-seq is really the driver of a lot of discovery, and we do multiple analytical approaches to this one data set. Finally, we do use the targeted ArcherDX assays of tumor RNA to identify driver fusions. And this is a billable test in our clinical setting at IGM that allows us to turn around information quickly, largely due to the fact that I mentioned earlier in terms of the large number of fusion drivers in pediatric cancer and the fact that many of these are now druggable as well. So of particular interest, I hope, to this group is really how we're doing all of the underlying computational analyses. And this is really played out here with the tumor versus normal enhanced exome sequencing along the top panel of the slide, and then the RNA-seq, which, as you can see on the far right-hand slide, invokes numerous different types of analyses, which I'll talk more about today. Central to all of this is the AWS serverless environment that allows us to literally stream packets of data off of the Illumina sequencers, do the BCL conversion in the Amazon Web Services cloud, and then at the end of that run, essentially fire off Docker containers with all of these different pipelines in them to effect the large-scale comprehensive analysis that I'll tell you about today. And much of the alignment processing in the cloud depends on our Churchill Rapid Aligner, shown here in reference from the original genome biology paper referring to that in 2015. So I'll talk about many of these aspects of the data analysis as we go through the presentation today. I also want to really point out that we depend greatly on data sharing, and this is a great example of web-based analysis at the Treehouse Childhood Cancer Initiative, run out of the UC Santa Cruz lab of David Hausler and overseen by Alina Vasky. We utilize this database in a multitude of ways, and I'll try to illustrate those today, most effectively through the UCSC tumor map that allows us to map these samples onto known expression profiles for patients and to interpret them accordingly. And I'll show you an example of that. So I mentioned the importance of fusions, and this is just our fusion detection pipeline. Because when we turn RNA-seq into a clinical profiling approach, we want to begin with a CAP-validated fusion detection pipeline. Because of the prevalence of these fusion drivers, we've effectively come up with the approach that is outlined on the figure on the right-hand side of the slide. The bottom line is that there are multiple fusion detection approaches. You can see 6 of them listed here that we're using. They all have strengths and weaknesses. And at the end of the day, there are 2 important points for clinical detection. First of all, minimizing false positives. We don't want to have too many results to look at. And then eliminating false negatives, because of course the worst thing clinically is to miss something. And so because we can't really encompass all of those want-to-haves in a single algorithm, we've chosen this approach to run multiple callers and then to evaluate the overlap calls as a secondary analytical step using this overlap script that's shown here. And then the downstream prioritization occurs that uses filtering to take a look at several databases that we've built that eliminate known false positives and evaluate read support, and then compare these to known fusions and their fusion partner genes. And then finally, there's a whitelist comparison that rescues known fusions that fall below the parameters of read support so that we don't miss anything that's already been reported. This just shows you where we're at in terms of about the first 100 cases that we evaluated. You can see the different fusions listed here along the bottom, and the height of the bar is the number of cases in which these have been detected. Many of these are known fusions, but some of them are actually novel fusions. And all of the fusions shown here are confirmed by our CAP-CLIA assay, so they can be written into the patient's medical record and used for care if needed. We also do a lot of work around gene expression outlier analysis. Part of this is using this Treehouse cohort that I mentioned earlier, but we also do comparisons to our own patient cohort. Here we're just running the RNA-seq data analysis using Salmon to obtain TPM values, and then we compare all the protein coding genes in a quantile normalized fashion to calculate their differential expression per patient. In some cases, we calculate the Mahalanobis distance and get a chi-squared p-value for the comparison, and we can also calculate v-scores from these log 2-fold changes. and get p-values accordingly. Finally, we want to assess drugability using drug-gene interaction database or evaluate the pathway by IPA. This is just a common output expressed genes. It's quite noisy and busy, as you can see, I guess, not noisy. But what we do from this is we evaluate these automatically through DGI and then focus in on the druggable targets. Here's an example as shown for a patient comparing to Treehouse. This patient has Ewing sarcoma, and you can see that the genomic alterations are in JAK1, this particular druggable target here, are quite high and fit well within the realm of other Ewing sarcomas according to the Treehouse. And here you can see on the right-hand side, this patient's JAK1 expression in red compared to other patients from our cohort evaluated through a DE-seq and showing that there's quite high expression here in non-CNS tumors compared to heme, which is the other tumor type in which JAK1 expression is identified. So this just gives you an idea of how we can evaluate what we think is overexpression in a contextualized way using the Treehouse or our own cohort. And then in many cases, passing these results forward, for example, to an immunohistochemistry pathology-based stain to truly validate them so that they can be written into the patient's medical record. Another way that we're evaluating RNA-seq is in the evaluation of the tumor microenvironment, namely what are the immune cells that are present, and using CyberSort and more recently CyberSort X as well as Timer, we're able to get essentially an evaluation of which immune cell are present, and in a case that I'll show you later, whether that immune cell profile is actually changing in the transition from a primary to a recurrent cancer. And then, of course, one of the things you may know from our work is the ability to do neoantigen prediction. We recently published an update to PVACtools, this 2016 paper from Genome Medicine, in cancer immunology research earlier this year for the PVACtools data tool set that's shown here. This is a highly modularized approach with flexible components that can all be run together or independently. There is a PVAC API, which is an open API REST interface. And we also have PVACviz, which is a browser-based GUI that allows one to put in very simple pieces of information from the patient's molecular profiling, DNA and RNA, and then evaluate the output of neoantigens. In the next example, I'll show you, we've actually used the PVACfuse module here to evaluate the potential for fusion drivers in pediatric cancer to be neoantigens. And this is work from Katie Miller, my postdoc, shown in the lower right-hand corner, where we've just taken a subset of different diagnoses from different patients where there have been identified gene fusions, and where Katie has evaluated the fusion junction in the reading frame between these fusion partners and come up with high affinity neoepitopes in many of the cases. So this is just one way of taking a different approach to neoantigen prediction besides the common approach, which is just to look at non-synonymous substitution mutations. Finally, just to wrap up the series of assays, we have been using increasingly the methylation arrays from Illumina to provide classifier-based subtyping. Here we utilize the classifiers that are provided in the public realm by the DKSC over in Germany. And in particular, this is important information to our neuro-oncologists who in many ways rely on the output from the CNS classifier to provide a tumor type or subtyping refinement. This can also provide insight into indeterminate diagnoses where the pathology approach is not clearly identifying the tumor type. And this can also tell us information about copy number, which we also get from the tumor normal comparison in sequencing, as well as the MGMT promoter methylation status. We're also planning to develop and add on the sarcoma classifier to this as well. So just to finish up then, I'll just give you a snapshot of where we're at with respect to patient enrollment. You can see this has been increasing over time since we opened this protocol in January of 2018. And this is a little bit out of date. We should have another month in place here soon. But as of this calculation out of REDCap, we had sequenced through about 152 patients so far across all histologies. This summarizes the impact of that sequencing, where on the balance, we can return at least one diagnostic, prognostic, or therapeutically relevant finding for 93% of our patients. Some patients actually achieve more than one, but at a minimum, one relevant finding is coming out of 93% of the patients that we've studied so far. And you can see in the gold boxes here along the bottom the different types of findings that we are producing from this combined tumor normal and RNA-seq based profiling. So I want to turn my attention now to just a few brief case studies to give you an idea of how all of these parts and pieces work together to bring genomics into the practice of precision cancer medicine. I'll start here with this case study in a very young child, initially diagnosed at age 1 in 2013 with this meningeal sarcoma. You can see on the far left in the MRI that was removed by gross total resection, and the postoperative MRI illustrates that. The patient went on to receive proton therapy, and this sample was sent out to a commercial sequencing entity for evaluation, where there were two findings. One, a quote pathogenic mutation in an intestinal cell kinase, and two, a noted variant of unknown significance in PDGFR beta. So remember those results because they'll become important here in just a minute. We studied this patient upon recurrence at age 5 in 2018. Again, a gross total resection, and in this case, the patient was enrolled onto the IgM cancer protocol and studied. What we identified is shown here on the left-hand side. I mentioned the intestinal cell kinase. In fact, this is a germline alteration. It's a 12 base pair deletion in this gene and is a unknown significance, although may have something to do with the suspect cancer susceptibility in this family because the patient's father actually passed away early from colorectal cancer. But we didn't have the father's sample to test, unfortunately, because of his untimely death. Importantly, the previously described VUS in platelet-derived growth factor beta, shown on the right-hand IGV plot, is in fact a 15 base pair deletion, and interestingly occurs in this juxtamembrane segment of the protein structure, which is shown on the far right. This is a well-known deletion that essentially turns off the autoinhibitory domain. And so the protein with this mutation in place is effectively firing at all times. This is a predicted gain of function mutation in this receptor tyrosine kinase. When we look at the corresponding RNA expression of PDGFR beta that results from this deletion that I showed you, you can see that the patient has highly elevated expression of the RNA. And indeed, from an ingenuity pathway analysis, you can see quite clearly that this PDGFR beta is at the axis of a very highly elevated and activated cellular pathway that's going on to drive this patient's cancer. The good news is that there are therapies for PDGFR beta, many of which are shown here. This is, as I mentioned, a receptor tyrosine kinase. And so there were a variety of different targeted therapies available for treating this patient. The oncologist in this case decided upon dasatinib, and we can see the net result showing here from the most recent scans on the far right from this patient. He continues to be alive very well without deficits from the neurosurgery and subsequent treatment and without any evidence of disease. Here's yet another case now combining information about the immune microenvironment that I described earlier and one of the areas of my own specific research interest. So this is a case of what I call a longitudinal sample, where we had both the primary and the recurrent cancer available for study. This is in patient JL36, a 10-year-old male with a recurrent CNS cancer. You can see that the pathology diagnosis is a bit different from 2015 to 2017, and that it was even sent out to St. Jude, to David Ellison, and categorized even differently. So this is what we often refer to as an indeterminate diagnosis. And as I mentioned, we studied both the primary and recurrent tumor. The major finding in this patient was an EP300 BCOR fusion. We confirmed this clinically after we identified it with that ensemble fusion detection approach that I mentioned earlier. And we proved it was present in both tumor samples. So this is an original driver event in this patient's cancer. We then went to search through the literature to identify other instances of EP300 BCOR fusion. This was a couple of years ago. The only thing we were really able to find, shown on the top panel here, was this oligodendroglioma from TCGA that also had a EP300 BCOR fusion, but with slightly different breakpoints than in our patient. So this remained a bit of a mystery in terms of what we might be able to do with the information. But at the time, we went forward to look further in the literature and identified this paper showing that a subset of 10 patients with CNS high-grade neuroepithelial tumors actually had an internal tandem duplication in BCOR. And from this and several other similarities that I don't have time to go into, we hypothesized that this translocation was biologically similar to these ITDs and might represent an alternative mechanism. One of the downstream manifestations of EP300 BCOR fusion shown here is our patient's two tumors, the primary on the far left, the recurrent more intermediate in terms of expression of epidermal growth factor receptor, which is of course a druggable target with a multitude of different drugs available for it. And in the fullness of time, when we needed this information clinically for the patient, we were able to identify this newer paper from early 2019 showing, quote, recurrent EP300 BCOR fusions in pediatric gliomas. So when I say quote, literally, this is 3 patients that were identified across several different institutions that satisfied the definition of recurrent, and basically are shown here with different, 3 different cases with 2 similar and 1 different BCOR, EP300 BCOR fusion. So our patient reflects the 4th known case as such, but nevertheless, in this particular paper, EGFR overexpression was also identified, and this remains a therapeutic modality that is now being trialed in this patient under progression. I wanted to delve a little bit deeper into this patient before I finish up with the last case and just talk about these longitudinal samples. So what I've presented on this slide is a series of 3 different pieces of evidence. On the far left, estimate analysis showing that in the primary tumor, the immune score is negative, but it's very highly positive in the recurrent. In the cybersort analysis shown in the middle, you can see the primary on the far left and the recurrent sample in the middle of this display, showing that there's a p-value significant result for the recurrent cancer with a large infiltration of M2 macrophages as well as monocytes. And that in the nanostring immune typer approach, we also look at essentially a dearth of immune cells in the primary here in blue, lower down, and then higher up in the recurrent cancer, a multitude of different immune cell types shown at high significance from the Z-score that's calculated by the software here. So this seems to be not just for this patient, but for many patients in the CNS setting, a reproducible phenomenon where we see an increase in immune infiltration in the course of recurrence. We evaluated this a little bit more by looking at neoantigen load. Here on the left, you can see the difference in the number of mutations identified in the primary on the x-axis and in the recurrent cancer on the y-axis. And then when Katie went through and evaluated neoantigen potential for these mutations on the right-hand side of the slide, you can see that in the primary tumor, really only one neoantigen achieves significance, whereas in the recurrent, there are a multitude more. And this is often the notion of why we get increased immune infiltration in the recurrent setting is because of the increase in the number of neoantigens. Some of this work has gone successfully forward to gaining new funding for our group, as well as several other investigators here at our children's hospital. And this recently awarded NCI funding under the Cancer Moonshot will now attempt in the preclinical setting with mouse models to evaluate different ways of impacting CNS as well as sarcomas using different immune modalities, such as shown here, natural killer cells, oncolytic viruses, targeting myeloid cell-based immunosuppression. And we will be using all of the comprehensive immunogenomics, some of which I have time to talk about today, some of which I don't, to address and study the changes that these different modalities bring to the tumor immune microenvironment in these preclinical models, and hopefully moving forward into the clinical trial setting. So I'll just finish with one last case, which is fairly recent to us, but shows that even in the pediatric cancer setting, we do get exceptions to this rule of low tumor mutation burden. And this is a patient with Lynch syndrome associated recurrent glioblastoma. Here's the case history for this patient. You can see a 2019 presentation in September. The diagnosis is down below, high-grade neuroepithelial tumor. You can see that post-operatively, the patient looks pretty good in terms of the gross total resection, but just a month later on follow-up imaging has an extraordinarily large tumor mass that's recurred in the original site. This patient then went on to focal radiation and veliparib, followed by a course of temozolomide, but in early January of this year on follow-up, still had a very large and increasing tumor mass. And so this patient was enrolled onto the IgM cancer protocol to study this recurrent cancer. And what we were able to identify from the germline analysis is shown here. This is a patient with an inherited PMS2 pathogenic founder mutation, which is associated with Lynch syndrome and a significantly higher susceptibility to cancers. The patient also had a polymerase epsilon variant, but this was of unknown significance in the germline. What we see here is that there are a variety of monoallelic germline alterations of mismatch repair genes that are associated with Lynch syndrome. And these give increased risk for cancers, as I mentioned, including brain cancers. And biallelic germline alteration would be associated with constitutional mismatch repair syndrome. But this patient just had a single PMS2 pathogenic variant in the inherited genome. The second hit is in the somatic genome. You can see here at the top, this second hit is a biallelic, results in biallelic inactivation. This is a frame shift variant. And secondarily, a POL-E known mutation, but not known what combination would be here with the rare germline POL-VUS. You can also see that there are a variety of other mismatch repair genes that have alterations that have been detected in this patient's genome. And that's not terribly surprising when you look at the fact that this is an ultra-mutated tumor. There are over 14,000 pass filter variants that are detected in this patient's exome. Some of which are in cancer genes, again, above and beyond mismatch repair, as you can see on the chart below. And were also validated with immunohistochemistry in some cases. We did pass this patient sample forward to OSU, to the adult hospital, to look for and to verify that the PMS2 variants had indeed eliminated the expression of PMS2 protein in this patient's tumor. So this is now a patient who will qualify for immune checkpoint blockade. I'll just show you one last example here of the DNA methylation analysis. The t-SNE plot is on the right-hand side, and the arrow points to this queried sample, which maps directly together with a large grouping of glioblastomas, IDH wild type, and classifier shown here. So this is now the completion of this patient's case at this point in time. We are following up by treating the patient, as you can see here, and as I mentioned, with a checkpoint blockade inhibitor based on the FDA approval of these types of drugs for patients with high ultramutation tumor burden. And this will also, because of the PMS2 genetic variant, indicate testing for other family members, and of course for this patient, heightened surveillance. So just to finish up, hopefully what I've been able to show you today is that these clinical genomic assays of pediatric cancers are really adding important clinical evidence. They impact treatment planning. They sometimes change or confirm diagnosis, and they provide prognostic information. We really feel it's important to combine the data from DNA and RNA to give this information about cancer drivers, the alterations in gene expression that may be epigenetic in nature, as well as the immune microenvironment as we move more into an era of immune therapy. And we've also contributed significantly to these efforts to share data, such as by contributing our RNA-seq data to the Treehouse. We're trying to support the use of these internet-based analytical and decision support tools. Because of the rarity of pediatric cancer, the importance of data and tool sharing is really critically a part of what we're doing now. And I'll just finish by saying that it takes a very large number of individuals to make this engine work. They are shown here, including our important clinical collaborators who are identifying these patients for us and helping us to better understand our findings in the context of the patient care. I do want to also acknowledge external collaborators shown here, Obi and Malachi Griffith and Jasreed Hundal for the PVAC work, Olena Baskey and David Hausler for the Treehouse project, and Bob Schreiber, with whom I work closely on other immune-based modalities. And finally finish with a special thanks to our patients and families and to you for your attention. And I'm happy to take any questions at this time. Very much.
Thank you, Elaine. Great talk. There are a number of questions on the Q&A board, but before we get to those, I'm going to use the moderator's prerogative and ask you a few questions myself.
Okay.
And so what's the, so one of the challenges in computational biology is the development of the structural variant tools and prediction from next-gen sequencing data. And so what is the concurrence of the RNA fusions with structural variants sort of that would match those fusions? Do you know those numbers?
Yeah, I think the concordance is typically pretty high. I'll tell you that at one level, we sometimes do have cytogenetic data that's been produced, or in some cases, microarray data that's been produced for some of these patients. I will tell you that in the case when we identify a fusion candidate, often the first step in evaluating that fusion candidate from RNA-seq data is to go back to the IGV evaluation of the fusion partners. And of course, you could typically guess that in many of those cases, we're identifying the mismapping of reads that are indicative of where the fusion has occurred. So we typically do use that as a part of the confirmatory work, but we're not using what I would call the typical approach that's been used in the past, which is sort of look at DNA first with a variety of tools and then look at RNA. For us, it just works better to go back the opposite way.
Okay, so looking at the Q&A board, and Elaine, we're going to do a few of these for the next few minutes, but if there are some left, we really would love it if you could go answer, answer some answers on them. So the first one is the, why are you doing exome versus whole genome sequencing?
Yeah, no, that's a great question. And I think it really comes down to two factors. One is we feel like that combination that I talked about of 30 kb resolution probes gets us what we need outside of the exome for copy number altered regions, including these in many cases, highly prognostic chromosome arm level deletions or amplifications. The other reason is because of depth. And, yeah, it's true that the cost of whole genome sequencing has fallen over time, but really at the depth you need on some of these tumors, I mentioned that we're aiming for 250-fold. That's a reasonable average, but generating 250-fold depth from whole genome is still a pretty expensive enterprise. So I felt like the enhanced exome approach that we adopted is kind of like the same thing. We're getting the information that we need in terms of copy number, but we're not paying that price for 250-fold or higher whole genome coverage on the tumor. So that's really where we're at.
Okay, so the next question from Selkan Aiden is, how are you utilizing DeSeq when there are many points of comparison? Are you using an alternative tool to identify differentially expressed genes where you can't do pairwise comparisons?
Well, we're not doing pairwise comparisons per se. These are usually, the reason for DeSeq is because, and also now we're starting to look at single sample gene set enrichment analysis as a maybe alternative to DeSeq. We're typically looking at these patients in isolation, right? And we don't have the luxury of adjacent normal tissue. That's really an anathema in many ways in the clinical setting because you can't take additional brain tissue, for example. So in one sense, we're looking at the comparison to Treehouse, which I talked about, because there are increasing numbers there of patients with similar diagnoses, less so for very, very rare cancer types. In the CNS setting, we're also getting great, help, if you will, from comparisons to the BrainSpan database, which is single cell data coming out of fetal tissue and developmental tissue in many cases, which allows us to look at that very young brain RNA expression profiling. So we do tap into that database. And of course, with the incredible work being done in the Human Cell Atlas, I think over time we will really have a very comprehensive single cell based analysis capability. But then GTEx is also something that we look at. So it's really always the patient in comparison to lots of other data, but not to themselves, just to be clear.
Sure. So the next question, I'm going to sort of tweak it a little bit with and add in my question related to it as well, is with respect to patient privacy. And Zento asks about the patient privacy being preserved on AWS-based computational analysis, but also, is it harder to get consent from families when children are involved? Is the whole children versus adult cancer complication?
Yeah, so these are great questions. I will say, first of all, that we have fully documented over a year's time the HIPAA compliance of our Amazon Web Services Cloud to the satisfaction of our hospital, IS. So that's all been verified as HIPAA compliant. And when we originally approached the hospital about this, there was the same sort of knee-jerk reaction. And over time, we were actually able to show that probably on the balance, the HIPAA compliance in the cloud is much better than it would be in on-prem computing. So yes, I think we were able to sway that. And even the NIH has approved that to some extent. So I think we can't be terribly wrong. In terms of consent, it's a little bit tricky. So I'll see if I can explain this briefly. For very young children under the age of 8, we approach the parents for consent. And we walk them through all of these aspects of data privacy, the possibility of a data breach, but the need to have a second set of data to really verify that it's that individual. I think the real sensitivity in pediatrics is that some of these cancer types are exceedingly rare. And so you may be able to even identify, so to speak, a patient based on that very, very rare diagnosis. But we do have what's called a patient asset, ASSENT, assent, that is for kids between ages 8 and 16. Above age 16, the patients themselves can consent, and then the parents as well. And I think the thing to keep in mind, I didn't mention it, is that we are, when possible, collecting at least a saliva sample for mom and dad. We don't often study that in the context of focusing on the patient, but in cases where there is some germline susceptibility that's been, predicted, we can go back to those samples from saliva and just do a spot check for that variant. And oftentimes that's very important information to have, of course, for siblings and for genetic counseling, et cetera.
I think we have time for a few more. So Lord Penna asks, how do known false positive gene fusions, how are they determined, I guess?
Yeah, so they kind of come in different forms and flavors. One of the first things we do is just look at the positioning of the predictions. A lot of times the false positivity comes out of genes that are essentially run on transcripts. So if they're on the same chromosome and proximal to one another and in the same direction, if you will, on the chromosome, then those are typically quickly ruled out as false positives. There are a few other examples, but essentially what we've done is over time in running this, this prediction approach that I talked about, we've been able to come up with recurrent sources of known false positivity, and we just build those into that database so that we don't have to have a clinical director or a variant analyst looking through it. We can just parse through it ourselves automatically, if you will, as part of a script approach. To compare the alignments for that patient's data to known alignment sources of false positivity. So that's probably the easiest way to rule out a lot of these false positives. And like I said, many of them come from just run-on transcripts.
So I'm going to sort of tweak a question from Jean-Christophe Lachance and ask you to comment on sort of the timing, the time of processing the data. And the various tools and getting it back to the patient in time for the appropriate intervention. Yeah,
super important question. Our overall turnaround time is typically on the order of 2 to 3 weeks. I think a lot of what helps this is the Amazon Web Services. Of course, you can spin up as many instances as you need, essentially, and then bring them back down again once that initial data crunching is done. Probably the biggest CPU hog is that 6-caller fusion pipeline that I showed you. And we will work over time, if you're alarmed by that, to try and reduce the number of callers. But right now we're comfortable with where we at. We started with 7, we eliminated 1 over time, just because false positives. And some of those are better computationally in terms of speed than others. So that's really the rate limiting step, I would say. But in general, then it's another week for the analyst to go through the data, really hone in on what's important, do that reach out to the literature that I mentioned in terms of, has this been seen before, that sort of thing. And then put together the report that we would then present at tumor board. So I typically show a picture of us at tumor board on Friday mornings is the CNS tumor board. We also do the sarcoma tumor board and then the generalized oncology tumor board. So 3 per week, where we're typically presenting 1 or more patients with our findings. Coming out of that, because we're still in the research setting, to be clear with this protocol, we would then get the physician to say, yes, I need that information to be verified, confirmed in the CLIA lab so that we can write it into the medical record, and I will use that moving forward. And of course, because at IGM we have this research part and this clinical part, we can put that directly into our clinical lab and get the confirmation if indeed it does confirm. So that's really the setting that we're doing this. And like I said, we're trying to return things in about 3 weeks on average. That works for most patients. I will tell you that we have had patients like 2 weeks ago where we really needed the information as quickly as possible. We got the sample in the door on Monday, and I was working on the 3rd of July, which with many of my analysts, which was supposed to be a holiday, but trying to get this vitally important information back to our physicians as quickly as possible. So that's about as fast as it can happen. But we can, you know, I think the 3 weeks is satisfying to the provider so far.
But I mean, I guess it's, you don't talk about the bad stories, but presumably sometimes it's too slow as well. There have been cases.
So this is a great question, and I will tell you that we're not perfect on that, but one of the questions that we ask on that initial REDCap form is what's called a Lansky performance status, which is specific to pediatrics, but is really essentially how well is the patient? And then we also have another question that says, is this patient expected to live longer than 3 months? And so, you know, assuming that those get filled out correctly, sometimes things happen. I'll tell you, just patients, you know, go downhill much more quickly than we anticipate, than our physicians anticipate. So I think on the balance, between those two questions, that's a consideration of, is a patient too sick to really benefit from this approach? So we try and limit that as much as possible.